And so this is just a little chart that diagrams which amino acids correspond with the codons that encode for them. So just a little side note, gene sequences can be thought of like as a sentence. So if the whole sequence of the gene is a sentence, then each of the units in the sentence would be like the words. So the words of the gene sequence would be the codons, or the three-letter sequences. And each of those codons is then going to be what gets translated into the amino acid. So the codons are really important for making sure the specific amino acid that they're supposed to encode for gets put in the correct order within that polypeptide chain at the end. And this table is just an easy way to figure out which of the three-letter sequences corresponds with each of those amino acids here. And so since each codon has 3 bases, there are going to be a total of 64, so 4 because there's 4 different nucleotides, and 3 because there's 3 different bases that can be placed in it. So 4 to the 3rd power is going to be 64. So there's going to be a total of 64 codons that can be encoded for. And all of these codons are written in the 5' to 3' direction. We should also note here that 3 of the codons on this table are going to, they say stop, and this is going to be the stop codon. So when there's a stop codon, all it's going to do is just terminate translation. So it's going to be telling the ribosome to just stop translation there. So the 3 stop codons are UGA, UAA, and UAG. So a fun little acronym for those are, for UAA, it's you are annoying. For UGA, it would be you go away. And then for UAG, it's you are gone. So just an easy way to remember the 3 of those. It's also important to note that because every eukaryotic protein starts with the exact same amino acid, methionine, the codon for methionine, which is AUG, is going to be kind of considered like the start codon, since it's going to be starting all of those sequences. And just to reiterate, each codon here on this table, each codon is only going to encode for 1 amino acid, but every amino acid can be encoded by multiple different codons. So even though each of these codons can only result in 1 amino acid, the amino acids can have a bunch of different codons that sort of make it or encode for it. Okay, and so here we're just going to talk a little bit about mutations that occur. And you can kind of think of like genetic code as being degenerate, and that's because more than 1 codon can specify a single amino acid, like we said before. And all amino acids besides methionine and tryptophan are going to have multiple codons that encode them. So like if we look at that last table, you can see that the first 2 bases are usually the same for each of these amino acids. So like if we look over here, it's serine. So in the first position, it's U for all of these. In the second position, it's C. And then in that 3rd position, we have a little bit of variability here. So it's really the first and the second position that make sure that the amino acid serine gets produced. We can see something similar here for proline. So for proline, it's C in the first position, C in the second position, and then the 3rd position doesn't really matter. All of those 4 nucleotides are still going to produce a proline as long as the first and the second position are C. We see something similar here in leucine. So that's kind of an important thing to note because that 3rd position the 3rd base is going to be variable. And because it's variable, we call it what's known as the wobble position. Basically, it just gives a little bit of room for some error within that. And it's considered like an evolutionary development that was probably not designed, but like developed over time to protect against mutations that occur in the coding regions of the DNA. So therefore, if like a mutation occurred in that 3rd spot, the wobble position, they're usually going to be silent or degenerate, which basically just means that there's going to be no effect on the expression of the amino acid. And then subsequently, because that's like silently passed down and it doesn't result in a altered amino acid being placed there, it shouldn't have an adverse effect on the primary sequence of the protein. And so the next mutations that we're going to discuss a little bit here are missense and nonsense mutations. And so if a mutation occurs and it affects one of the nucleotides in a codon, it's going to be what's known as a point mutation. And, excuse me one second. Point mutations can be silent. So like in the case of the wobble position that we talked about before, that was an example of a silent point mutation. Or they can have more serious effects, which usually depend on where the mutation occurs. So like if one point mutation is going to change the amino acid that gets ultimately placed into the polypeptide chain that results, you're going to have a more serious effect than like if a point mutation just encodes for the same exact amino acid that it would have if the mutation hadn't occurred. So that's really important to note, that it kind of depends exactly where that mutation is going to occur on how serious a result it's going to have. And when the point mutations do affect the primary acid sequence of the protein, they're going to be what's known as express mutations. And expressed point mutations are going to fall into one of two categories. So they're either going to be missense mutations or nonsense mutations. And so missense mutations are where one amino acid substitutes for another. And then nonsense mutations are going to be where the mutation ultimately results in the codon now encoding for a premature stop codon. So that's going to have a much more serious effect usually than the missense mutation, because in a nonsense mutation, if the mutation results in early termination, it's probably going to result in a non-functional protein. So this missense mutation can also be referred to as what's called a truncation mutation. So the next type of mutation that we're going to talk about a little bit is the frameshift mutation. And so frameshift mutations occur when some amount of nucleotides are either added or deleted to a section of the mRNA sequence. And the reading frame is the name that's given to the 3 nucleotides of a codon. So it's sort of like how it reads it and the space that it reads it at. And when insertions or deletions occur, that reading frame can be shifted, right? So if you think about like the 3 positions that we had for the codon, which is the reading frame, if you're going to add a nucleotide in there or take one out, it's going to shift the whole line either down or up. And so this is going to have more than just like one consequence. It can either affect things that are upstream or downstream from the sequence as well. And that can also result in like the insertion of a premature stop codon or something more serious. So unlike point mutations, frameshift mutations usually result in a shifted reading frame, which are more serious consequences. And this also depends heavily on where within the DNA sequence the mutation is going to occur. And an example of a frameshift mutation is cystic fibrosis. So in cystic fibrosis, a deletion is going to occur at codon 508 of the polypeptide chain. And that codon 508 of the polypeptide chain is the region that encodes for the CFTR chloride channel gene. And when that deletion at 508 occurs, what results is the loss of a phenylalanine residue. That phenylalanine residue is really important for making sure that the channel is functional. So what's going to happen is a defective chloride ion channel is going to result. If you have a defective chloride ion channel in the cells, it's also going to lead to blocked passage of salt and water into and out of the cells. And we can think of a lot of ways that can be pretty serious. If water can't pass in and out of the cells and salt, the body's going to have a really hard time like maintaining that balance. So this usually presents phenotypically in patients as like an abnormally thick or sticky mucus that either lines their airways, but it can also line their organs. And that can pose a lot of serious health consequences for people who do have cystic fibrosis. But that's sort of like the characteristic way that people recognize cystic fibrosis.